Meiosis is the process of preparing gametes for reproduction. So while it is a type of cell division, we are actually going to focus on the production of a different type of cell, genetically different from the parent, so that each cell is prepared to either become an egg or a sperm. We've talked about homologous chromosomes before. We focus on the idea that these homologous chromosomes came in two pairs. You had one set from mom and one from dad. What we didn't tell you before is that because you have two sets of chromosomes coming from mom and dad, this actually makes you something known as a diploid organism. It means that you have two copies of each chromosome set, or we write that you are a 2N organism, N being chromosome set. So having two of those, one from mom, one from dad. Now, when we look at homologous chromosomes, we have talked before that these homologous chromosomes in these pairs are very important from the standpoint that they are similar but not identical. We talked about genes and how mom might tell you that you're going to have brown hair, but dad is going to maybe tell you you're going to have blonde hair. And those are what we call alleles or gene variation. And we use that so we are introducing a higher level of variation to the human population. But what we didn't mention to you is the fact that these genes actually have an address, so to speak, on each of your chromosomes. They're called a locus. This location or address associates chromosome number 1 through 22 for you. Remember, the 23rd set is X or Y. And then we look at a position on that chromosome. So maybe the pink end is 1, then 2, 3, 4, 5, and so on. Now, I think of this like houses on a street. If you're getting chromosome 1 from mom and chromosome 1 from dad, what we're interested in is the gene that sits, in this case, at locus number 12. Mom says pink, dad says white. Now, I understand this seems kind of strange to think about, but we have to keep in mind that our goal with meiosis is the production of a different kind of cell, and that different kind of cell is a haploid cell. So understanding that we get different alleles, different varieties of genes from mom and dad becomes really important, because this is why you look different from your brothers and sisters. Okay? Because what we're going to do is produce these haploid cells. One will be an egg, one will be a sperm, and when we put them together, we get you through fertilization. Okay? And in that, you are back to being a 2N or a diploid organism. This is critical. If we had an egg that was 2N and a sperm that was 2N and we made you, you would actually be 4N. That means that you had 4 copies of every single chromosome. You would have 92 chromosomes. Remember, number of chromosomes is species specific. If you have 92 chromosomes, you're no longer human. That's a problem. So we take this original parent cell and we're going to go through a process just like we did before of interphase. So we're going to grow, replicate DNA, and grow again. And then we're going to divide the cell. But instead of dividing the sister chromatids like we did before, we're actually going to divide the homologous chromosomes. So in meiosis 1, we divide homologous chromosomes, and this is actually going to half the number of chromosomes. Why? If we have 46 chromosomes in you, and you do DNA replication, you end up with 92 pairs, right? Or 92 sister chromatids, those X's, right? Well, when we do this, we'll show you that we're actually going to look at these sister chromatid X's as being these homologous chromosomes, and when we divide them, we're going to send half of them to one side and half to the other side into two new cells. So if you take 1/2 of 2n, you get 1n. Then we're going to go through a second meiosis without interphase. So there's no interphase here. So there's no growth. There's no replication. There's no more growth. We're simply going to just divide the cells again, and this time divide the sister chromatids like we did before in mitosis. Now, when we end up, each one of these cells for you is actually going to have 23 chromosomes in it. The whole idea with meiosis is to decrease the number of chromosomes. So go from a 2n starting cell to an n cell, so that we can take n as an egg, n as a sperm, add them together, and get a 2n of you. We talked about interphase. We said that in order to start meiosis, we would do interphase. We'll do interphase whether you're doing meiosis or mitosis, it doesn't matter. You start with interphase, but rather than just dividing the cell once, you're going to divide the cell twice in meiosis. And the whole idea is producing 4 daughter cells that are haploid. So my product that I'm after in meiosis is 4 daughter cells haploid. In mitosis, my product was 2 daughter cells diploid, and I wanted them to be genetically identical. The haploid cells, I want them to be genetically different. This is good. This leads to variation in the population. This is a very, very good process for us to do this. So interphase as normal. The phase names should look familiar to you. Prophase, metaphase, anaphase, and we'll have telophase and cytokinesis. But instead, metaphase 1 is where things start to get a little wacky. Right away. Remember in metaphase 1 in mitosis, there was a single line of X's across that middle. You see now, the homologous chromosomes have actually paired up. So the red ones from dad, maybe, the blue ones from mom, it doesn't matter. Just to show you that they have paired up. And when they pair up, they form this thing known as a tetrad. Tetrad, tetra is 4. So you have 4 sister chromatids stuck together. When you do this, what we're looking to do is to separate then the homologous chromosomes. Remember, the reason we do this is to go from 2n to n. This is the phase. Anaphase 1 is where we decrease the number of chromosome copies. The other thing we need to think about that we'll talk about more in genetics is this idea in prophase 1 that this tetrad, you see the legs of these sister chromatids kind of overlap. This is important. This is something known as crossing over, or what some of you may know as recombination. This leads to high levels of genetic variability. This is very, very important in evolution, and we'll come back to that. Telophase 1, cytokinesis 1. The first time we go through this, splits out again. We're going to have a cleavage furrow, split out those two cells. And those two cells now, no interphase. There's no in between. Those cells go immediately into dividing again. And this time, this part is just like mitosis, but with fewer chromosomes for you. So in this case, we're looking at splitting out the sister chromatids because we have a single X line on the metaphase plate. We split out the sister chromatids. We go through telophase 2 and cytokinesis 2, and you end up with haploid daughter cells. So N daughter cells. We need 4 of them total. And they're going to be genetically different.